Hello everyone, my name is Sina Falakian and I'm a master's student at SFU. Today I'm going to talk about the subject of my master's project. So DNA replication initiates at the origins of replication. So at the beginning of this process, there are some proteins called pre-replication complexes. These proteins get binded to the DNA molecule and separate the two DNA strands. And the location where the DNA strands are separated is called the replication origin. And so the separated area between the two DNA strands is called a replication bubble. And the Y-shaped structure between the bounded and unbounded DNA is called the replication fork. So as the replication forks move away from the replication origins, there are free nucleotides in the DNA environment which start getting incorporated to the free DNA strands and create complementary DNA. So when the replication forks span the whole DNA, we get two daughter DNA molecules where each of them share one DNA strand with the parental DNA. In bacteria, we have a single origin of replication. However, in eukaryotes, because of there are multiple replication origins because of their longer genome lengths and smaller fork velocity. And also we, in bacteria and brewer's yeast, we can see that there's a sequence dependence between the origins of replication. However, in human, there is no sequence dependence have been observed in this process. So we can ask some question about the DNA replication process in human, like where are the origins located along the genome, and when do they fire during the replication, and how the replication timing is regulated in this process. So to answer this question, a new experimental technique called optical replication mapping is developed. In this technique, some fluorescent molecules, so some molecules that are made of a thymidine-like molecule attached to a fluorescent molecule are inserted inside the cell's nucleus. And as the replication forks move, these fluorescent labels start to get incorporated to the replication forks. Then after the replication process is completed, the DNA molecules are fragmented and linearized inside nanochannels. And in this stage, they will be ready to obtain the ORM images. In this image, you can see an example of an ORM image. So here, the blue is the DNA, and the red dots are the labels that are incorporated to the DNA, and the green dots are the restriction sites, which are molecules that get binded to specific sequences in a DNA molecule and can be used as a barcode to locate the position of the DNA fiber along a chromosome. So the optical replication mapping gives a single molecule high throughput data, which can cover the genome about 100 times. But there is a sparse labeling problem, meaning that whenever we have an origin, the labels that are binded to DNA around that origin are sparsely distributed, and it makes it hard to infer the location of origins from the label positions. Also, because of the small number of labels that can be inserted inside the nucleus, these labels are used up in the first 2% of the replication timing. So, which brings us to my project, which is that we are trying to infer the position of the replication origin and the replication kinetics using these early initiation events. So to do this, we first need to have a model for the label incorporation around the origin. So in the previous studies, exponential decay model was proposed for the label incorporation rate, and it seems to be in a good agreement with the inter-signal distance distribution. However, we have observed an issue with this model. So according to this model, the average number of labels around an origin should be about 50. However, we have observed that the number of labels on each, the average number of labels on each DNA fiber is about 5. So there's a 10-to-1 difference here that we are trying to explain. So far we have two theories for this. So one is that the labels can spread into the DNA fiber from the initiation that occur outside of the fiber. And we have sort of been able to rule out this assumption. And the other idea is that the initiation can occur in later times. So in the previous model, the assumption was that all the initiations occur at the beginning of the replication process, but we are trying now to incorporate the different times of the replication of the initiation events and create a model, a time-dependent model for the label incorporation rate. So to summarize, the replication kinetics in human genome is regulated by the spatiotemporal organization of replication origins. And the optical replication mapping, or ORM, is a new experimental technique that has been developed to find a spatiotemporal organization of these replication origins, and we are working on a model for the label incorporation in ORM experiment to use for inferring the ORM data. Here are my references, and thank you for listening.